Hello everyone, let us continue our topic of molecular biology, that is the search for genetic material. In previous one we have discussed about packaging of DNA in eukaryotes and all. To start with, nuclein, a acidic substance present in pus cell, was discovered by Friedrich Miescher, and Mendelian principles of inheritance both were discovered at almost at the same time, that is around 1869. But to prove DNA is the genetic material as we know now, it took many years, almost a decade it took, that is to prove DNA is the genetic material. That is, by the time 1952 it was proved that DNA is the genetic material. If you look at the time gap, that is 1869 to 1952, it is almost 83 years. You might ask question why it is. It is because there are many constraints, that is their microscopy was not advanced and there is no advanced instrumentation to prove that DNA is the genetic material. But to prove which molecule present in cell is responsible for inheritance of the character, the quest was raised or increased by the year 1926. Previous discoveries which was done by Gregor John Mendel and Walter Sutton and Boveri experiments and other scientists like Thomas Hunt Morgan and other scientists has narrowed, that means the work of these peoples has narrowed that there are materials which are found in the chromosomes of a cell which are located inside the nucleus of most of the living organisms. That was a very fine tuning experiments which were done by these scientists, which was narrowed the broad search for genetic material into the nucleus. That means next discoveries has started to search the genetic material which is present inside the nucleus. But which molecule is the genetic material? There are many molecules present in a cell or in a nucleus, like DNA will be present, RNA will be present, proteins will be present, along with carbohydrates may be present. Now, which among them is responsible for inheritance of character was not clear. So, many experiments were done in order to prove which molecule among these, whether protein, DNA, RNA or carbohydrate, is the genetic material. For that, many experiments were done, but here we are going to discuss only milestone experiments, that means which have contributed much in the search of genetic material is DNA. Among them, the first and foremost important experiment was conducted by a scientist, Frederick Griffith, in the year 1928, and that was popularly called as Griffith transforming principle. Griffith, he conducted experiments, series of experiments using a bacteria, and these bacteria were experimented on mice. So they may ask questions like which are the organisms used by Frederick Griffith in order to conduct transforming principle. It might be carrying 1 mark, 2 mark or multiple choice questions they may ask for competitive exams. So they will ask question like which are the organisms used. So you have to remember two molecules, two organisms, one is bacteria, it is known as Streptococcus pneumonia or Pneumococcus pneumonia, or else it is called Diplococcus pneumonia, and the second organism will be mice. So the two organisms used by Frederick Griffith in order to conduct transforming principle was Streptococcus pneumonia and mice. While he was doing experiments using Streptococcus pneumonia and mice, he witnessed a miraculous transformation seen in bacterial cells. The transformation means change. So if you do not catch those terminologies, so remember simple terminologies, that is transformation means what? Change. So you may ask question, change means how it is? Actually, it was a physical change. You can imagine if you got two heads, within one night. While you are sleeping, one head, but when you woke up in the morning, you got two head, means that is a miraculous transformation. Such miraculous transformation was seen in bacteria also. So what is that? That change we will be discussing later. So once we understood the morphology of bacteria, then we will understand what is that miraculous change which was seen by Griffith experiment. So to know that, let us know the structure of Streptococcus pneumonia bacteria. Streptococcus pneumonia is a bacteria which is known to cause a disease called pneumonia in many of the animals, including human beings. When this Streptococcus pneumonia is grown in a culture plate, you know that agar, that is agar, you know, from plant kingdom, from algae, Gelidium glacialaria, they will extract agar, and it is known to grow the organisms in laboratory. So when Streptococcus pneumonia is grown on a nutrient agar medium, some of Streptococcus pneumonia produces a smooth and shiny colonies. So here I am writing without any edges here. So smooth and shiny colonies it will produce. And these smooth and shiny colonies producing Streptococcus pneumonia is called a cystrain. So you have to remember, they may ask differentiate between two types of Streptococcus pneumonia, they may ask. So remember, when Streptococcus pneumonia grown on a nutrient agar medium, some of the strains produces smooth shiny colonies and they are called a cystrain. And when the same bacteria it is grown on a nutrient medium, some may produces a rough colonies which are having a edges. So here you could finely differentiate between the two. So rough colonies they may produce, and those rough colony producing bacteria are called R strain, which means what? In Streptococcus pneumonia there are two strains, S strain and R strain. What is the morphological difference between S strain and R strain? S strain produces smooth shiny colonies, whereas R strain produces rough colonies. That makes the difference between S strain and R strain. But there is one more very important difference between S strain and R strain, which is S strain of bacteria produces smooth and shiny colonies because they have a mucus coat which is made up of polysaccharide. We can call it as a polysaccharide capsule. So there are cell, and cell will be surrounded by polysaccharide capsule. But R strain of bacteria do not have such polysaccharide capsule. So only the cell is present. Polysaccharide capsule is absent in R strain. In S strain of bacteria, since it is having a polysaccharide capsule, they are called virulent strain. Virulent means what? Causes death. Any organism, if it is calling as virulent, causing death. But R strain of bacteria is avirulent because polysaccharide capsule is absent. So you have to remember here very importantly that virulent and avirulent is because of presence or absence of polysaccharide capsule. Now let us understand what is the series of experiment conducted by Griffith. Griffith, he took S strain of bacteria, that is virulent strain of bacteria, and he injected that into mice. So virulent means you are knowing that causes death. So what might have happened to the mice? Obviously it has caused death. So here I am writing dead mice inverted, so you know that how it will appear. So for that I am writing, so you can remember this. And in second set of experiment, he took R strain of bacteria and injected that into mice. Remember other mice, not the same one, mice. And that mice do not undergo death, that is the mice was live. So you know the reason. S strain is having polysaccharide capsule, that caused the death. R strain do not have polysaccharide capsule, so that make the mice to live. But later he took another modification in the experiment. Griffith was able to kill the bacteria by heating. So don't imagine, children, heating means it's not burning. So whenever killing means here within experiment it represents what? They'll take some liquid. In that liquid they will put bacteria and they will heat or boil it so that it will die. So if it is burnt, no ash will remain. That using that they can't do any experiments. So remember that. So a strain of bacteria was heat killed and it was injected into mice. So what observation you could have now? Obviously the mice was live. What happened here? While heat killing, the polysaccharide capsule was destroyed. So if there is no polysaccharide capsule, there is no death of the mice. Later, in another set of experiment, what he did is he mixed heat killed S strain of bacteria with live R strain of bacteria. So now these two are in a mixture. They are not separately injected. They are in mixture. They are injected into mice. So now give some work to your brain. What may happen to the mice? Whether it is live, whether it is dead. So in previous you have studied R strain do not cause the death, heat killed S strain do not cause death. So we will think that the mice has not died, but the mice has died. This is one very important observation done by Griffith. And another very important observation is that Griffith was recovered live S strain from the dead mice. What he injected, heat killed S strain and the live R strain. But what he obtained from the dead mice is the live S strain. And these are two are the very important observations from Griffith. So they may ask question, what are the two important observation? One important observation is mice was died and he could recover S strain, live S strain from the dead mice. So from this observations he came to some conclusion. That important conclusion is the R strain of bacteria has somehow been transformed, means changed by heat killed S strain. That means some of the molecule was retained after eating in S strain of bacteria and that have changed the R strain into S strain. How it is changed? Some of the transforming principle present in heat killed S strain. That is, what is it? We don't know. It may be protein, carbohydrate, or it may be RNA or DNA, anything. So somehow it is retained after heat killing also. That had made R strain to synthesize mucus or polysaccharide capsule. Hence R strain is converted into virulent strain, that is S strain. So that has caused the death of mice. So here there is no magic happened. That is R strain is itself is converted into S strain. That because S strain, some of the transforming principle from S strain has transformed the R strain into S strain. So, but by this experiment, it is not concluded that genetic material is what, whether DNA, RNA or protein, it is not clear. And that clearance we will got in further experiments which were conducted by other scientists. So, until then, wait for my next video. I hope you like it. Thank you.